Hey friends, how are things out there? Today we're going to talk about invertebrate nervous systems a little bit, and we'll see what we can make of it all. We're going to discuss a paper by Jarvis, Bruce, and Patel that was published in PNAS some number of years ago. It's a pretty good paper, and I picked it because it's only like 6 pages long. So yeah, you can catch up and read it and all that kind of good stuff and get all the stuff I'm going to gloss over today. Yes, Frontopia, it did start. Here we go. I hope. Oh, an ad. There's ads. Oh, well, okay. Yes, here we go. So let's talk about this stuff. So let me think. I have to do a few things here, a few housekeeping things. I have to go here. I have to click on this. Oh, I have to click on a little button here. And there we go. Okay. So what I'm talking about is some cool stuff on comparative development of the nervous system. And this is work that builds on a familiar story. So what we're looking at here is staining for the early gene expression in Drosophila. So what we see in this little diagram here is that there's a hierarchy of genes that are switched on to turn on positional features in the embryo. So we start with maternal factors that just sort of roughly lay out anterior and posterior. Then there is a set of genes that are turned on that mark off boundaries within that anterior posterior gradient. So this looks like a hunchback to me. No, that can't be hunchback. It's going to be Krupel. Anyway, there's a whole series of these that are turned on. They're just showing you one illustrated here. Then there are the pair rule genes like Eve and Futz, which we'll come back to. Futz is short for Fushi Toratsu. We'll come back to that. The pair rule genes are expressed in alternating segments. And then after the pair rule genes are expressed, we switch on the segment polarity genes, which are turned on in each and every segment. So like I said, a hierarchy. We switch these things on in order. Then there are also the homeotic genes that are expressed on top of this that define broad body regions. So for instance, UBX is a thoracic gene. If you've got UBX on, you're part of the thorax. Okay, so we got this hierarchy that switches on to impose order on the anterior posterior axis of the animal. The thing is, this is a really old hierarchy. So Patel knows quite a bit about crustaceans. So he's writing about crustaceans here. And crustaceans have these same or similar genes with similar sorts of roles. So what we're seeing here in red is the expression pattern for ultrabithorax in a range of crustaceans. So these are all crustaceans here. And what it's showing is that the domain of expression of UBX defines the domain of expression, the morphological expression of legs. And up here anteriorly, for instance, in the lobster, the two anteriormost segments do not have ultrabithorax expressed, and they form feeding structures. So Patel himself has done a series of experiments where they modify the expression of UBX, and it's beautifully correlated with the expression of feeding structures versus locomotion. So that's been done very elegantly, very thoroughly. Now, I'm just showing you this because I want to point out that there's a lot of deep homology here. So this is a couple of different arthropods. So there's an insect. This is an insect. So this was Drosophila. This is the grasshopper Schistocerca. This is a crustacean. It's a tiny little a little shrimp that infests aquaria. These are Spiders, yay! We got spiders today. There's a couple spiders there, and myriapods. So centipedes and millipedes. These are showing the expression of segment polarity genes. So they all have the same pattern. So this is a particular segment polarity gene called gooseberry, and it's in every segment in all of these animals, which tells you this is an ancient homology. Keep in mind that crustaceans and insects, these crustaceans and insects probably diverged about 500 million years ago. And insects evolved, what, 350, 400 million years ago. So we got big, big differences deep in history, and we still retain this pattern of expression. Now, what I want to focus on is this aspect of it. Okay, what we're looking at here is a cross-section through a typical insect. This could be Drosophila, it could be Schistocerca, whatever. And what you've got in these organisms is this distinct dorsal ventral pattern of organization. So there's a dorsal epidermis, then down here we have the neurectoderm. In insects, the ventral portion of this tube is going to form the nervous system and ectoderm, the cuticle of the animal. And right in here is mesoderm. Before this picture was illustrated, the neurectoderm and the mesoderm would have been continuous, but everything migrated down ventrally and the mesoderm folded into the interior of the embryo. It's going to form things like muscles and organs. And the neurectoderm fused at the midline. And this structure is going to form, at least this ventral portion is mainly going to form the nervous system of the animal. Now, how that works is, it's a little bit complicated. So you've got this sheet of cells that are going to form ectoderm, they're going to form glia, they're going to form neurons. And what will happen in the insect is that some of the cells will delaminate. They'll migrate into the interior. This is from Lewis Wolpert's textbook, and unfortunately he's drawn it upside down. I don't know why he did that. But remember, the insect neurectoderm is ventral. So this is the ventral side of the animal, and everything delaminates into the interior in a dorsal direction. So this is another comparison of the process. On the left, this is an insect, an invertebrate, and on the right is a vertebrate. And this is what most people are familiar with in the formation of the nervous system, is the folding of the neural tube. In vertebrates, the neural tube just it's dorsal, it's on top of the animal, and it folds around to make this elegant little tube. That's the neural tube there where all the nervous system is going to form. And in addition, this has domains. There's positional information in here. Insects, sort of similar in some ways, except of course it's ventral. Nervous system is ventral, so there it is down there. And then when you look in there, what you find is also domains of similar genes to what you see in vertebrates being expressed. You don't get the infolding of a tube. Instead, what you've got is that delamination where individual cells bud off and move into the interior to form the nervous system. Because you don't want your nervous system just hanging out naked on the ventral surface, right? You want a cuticle there, so they move inward to form the nervous system. Now, once upon a time, I studied this whole process years ago with grasshoppers, and I drew this illustration to sort of put it into 3 dimensions and see what's going on. In grasshoppers and other insects, you got this neurectoderm, and one of the cells will move into the interior and become something called a neuroblast. So a neuroblast is a precursor cell. It's going to eventually form glia and neurons. In Schistocerca in particular, it's really obvious, but it's also in Drosophila. That cell, that green cell there, goes through a series of unequal divisions and buds off a chain of what are called ganglion mother cells. And it's kind of cool to see in the actual embryo. You got these big cells down deep, and then you got these little swirly chains of ganglion mother cells coming off of them. So they form long lines, sort of a conga line of ganglion mother cells. And then these ganglion mother cells will eventually divide one more time, just one more time, and produce neurons in that segment. It's very stereotyped. That's the cool thing about it, is it's very repetitive. You can see the same thing going on in crustaceans and spiders, for instance, with some differences. In crustaceans, they don't actually bud off into the interior. They don't actually delaminate. They're going to form right there in that space, and then they're going to bud off daughter cells that form the nervous system of the crayfish or the lobster or whatever. Spiders are more like the insects in that they do delaminate, but they don't delaminate in single neurons or single neuroblasts that move in. They form these grape-like clusters of multiple neuroblasts that come out all at once to form the nervous system. Now, in Drosophila and in grasshoppers and in, all insects, it seems, if you look down on the nervous system, what you see is a characteristic pattern of these neuroblasts forming. They appear in 7 rows. There's 5 different layers here, and it's true for Drosophila and grasshoppers. Remember, half a billion years of separation, but they form these early neuroblasts that are recognizable. They've got names, well, numbers, and you can actually recognize the same neuroblast in segment after segment after segment, and in animal after animal after animal. So this was kind of mind-blowing to me when I first started working on grasshoppers, because I looked down there and you see this surface, it looks like a cobblestone road with all these little cells. And initially, of course, you can't tell where you are. It's just all these cells all over the place. But after you stare at them for a while, you start recognizing the pattern, and you can see these similarities in different segments, and you start to recognize the individual cells that are emerging there. And so I did this in grasshopper, and that's where I was studying them. And then one day I got some Drosophila and looked at their embryos, and it was mind-blowing because I could see the very same cells, the very same neuroblasts appearing in the fruit fly as well as the grasshopper. They were much smaller in the fruit fly, so I'm glad I was working on grasshoppers. Grasshoppers are big and juicy and easy to work with, but these were, you could just see the same pattern emerging everywhere. So they formed this initial network of neuroblasts. And remember, these are going to divide to make those ganglion mother cells, and then they'll make neurons. So this is just the foundation of the organization of the nervous system. Now we can carry this detail even further. So this is some work by Chris Doe. And this also kind of blew my mind, because when you look at these cells and you stain them for different transcription factors, different genes, what you see is a pattern. And it's a combinatorial pattern. So they're all expressing the same suite of genes in each one. Each of these neuroblasts is unique in their combination. And as you know, as the embryo gets older, you get more and more of these neuroblasts until you get this final pattern over here. But you can look in there and you can just see the individual neurons each have their own unique combination of genes being expressed, specifying their identity. Furthermore, this is the nervous system. All the work has been done in making a segmented animal already. So they've already made this segmental stripes. They've turned on the homeotic genes, all that kind of stuff has already happened. This is a little farther down the road. And what happens then is these neurons reuse all those genes. So you see the staining, the label here. So there's FUTS, for instance, Fushitoratsu. That's the pararule gene is expressed in every other segment. And now in the nervous system, in every segment, it's expressed in a subset of the neurons in here. So there it is turned on right in here. Similarly, there's some others like odd skipped, another pararule gene, wingless, engrailed, all of these really familiar genes from segmental pattern formation get reused over and over again in novel and interesting ways. In some of the ways were kind of unbelievable because these cells are going to go and form neurons, and then those neurons are going to send out processes, axons that branch and innervate different tissues of the organism. There are some of these pararule genes, like even skipped, Eve, is expressed again in the growing axons of the neurons. And most surprising of all, sometimes it's only expressed in certain regions of the axons. So there's this whole other layer of combinatorial coding that's essential for building a complex nervous system. Okay, as I said, I used to do this stuff. I used to work on this, and this is just to illustrate what it actually looks like. So you get all those nice diagrams with the little beach balls for the neuroblasts. When you're actually doing the work, this is looking at a grasshopper, a single segment of a grasshopper, and we're looking down on the one half of the nervous system, and you can see what it actually looks like. There's all these cells here. This is shot with differential interference contrast microscopy, so you can see the cell membranes here fairly well. And like I said, when I first did this, it was, oh my gosh, what is this? This is just a mess of blobs. And when you look at them for a while and you start to practice at it, you begin to learn, like for instance, these cells, this is the posterior corner cell, the anterior corner cell. There's another cell called MP1. I just went in with a paintbrush and in the computer and turned it blue. And this is Q. This is the cell I was most interested in. I colored that one orange. But you can just sort of see these coming together. And once you've got that mental search image, you can go in every segment and find these cells over and over again and do experiments on them. So I was doing experiments like here I've stained MP1 blue and Q red, and that's just to reflect the fact that I also did experiments where I stained them with lipophilic dyes. And you can actually see that they're very complicated cells with all kinds of processes all over. And this is what I was interested in mainly, is not necessarily neuroblast formation, although the stereotypy of neuroblast formation made it so easy to find the same cell in every single segment. And then, I was making time-lapse movies, like these are some, these look kind of grainy and crude, and that's because this was done in the 1990s. Yeah, we had to work on the technology there. These are all pictures of that Q neuron I mentioned, which grew out and crossed the midline, forming the posterior commissure of the animal. And it's got these really active filopodia that are just flickering in and out all over the place, which are fun to watch. So that's what I was doing, was looking at formation of the nervous system at the point when the ganglion mother cells had divided to form the neurons, and the neurons were all growing out and doing their thing to build a nervous system. Now, in this video series, I have the goal of keeping it fairly simple, so I'm going to stop right there. And let's see if we can get some questions here. If anybody's paying attention. Oh, Now I have to go and find that. There we go. We turn that off. We can turn that off. You can see my face, I hope. Yeah, there I am. Okay. So let me get into the Q&A here. We just got a few questions. Oh, there's Rory Katz. Yeah, back to you. Okay. Let's see. I think, yeah, gap genes is a class of genes, and it denotes things like hunchback and Krupel and so forth. So there's a whole battery of gap genes that together fill in the spaces of the nervous system and the other tissues of the embryo. Yeah, HB, yeah, those are abbreviations. HB is hunchback, KR is Krupel, GT is giant. Okay. So yeah, they do all this. And then Phrenotopia asks, is the ventral midline perhaps where the mouth zip together? No, probably not. This has nothing to do with the well, it does have something to do with the blastopore. But yeah, the blastopore is going to form anteriorly in these animals. And then you get this fissure forming the length of the animal. It is the place where gastrulation occurs. So now that you're looking at me, I can do hand gestures. Okay. So what you've got is this tube-shaped animal like this. And ventrally, everything's going to migrate down, and then it's going to roll into the interior. So the stuff that is most ventral along the midline of the early embryo is going to form the mesoderm. Okay. And the stuff just lateral to that is going to form nervous system. So it's going to just do that roll inward, make an interior mesoderm, and then the ventral region is they're going to link up and form a continuous nervous system. Okay, Fergus asks, yeah, you don't believe this, do you? Yeah, it's the same genes and the same proteins. That these genes are pleiotropic. That Eve, for instance, even skipped, is important in pair rule formation, so it's important in segmentation. Then it gets reused, that very same gene, the same proteins, in specifying cell identity within the nervous system. And then it apparently plays a role in the formation of neuronal pathways, where it's expressed in some pathways and not others. Yeah, it's a marvel of efficiency that all these genes get reused in different ways within the development of the nervous system. So you ask how. I don't know how. I would like to know how. There's some magic going on there. I don't know how it works, but yeah. Okay, any other questions? I am not averse to keeping this short and simple. I'm actually thinking about talking more about the nervous system next week if people are interested. So any other questions? Okay, well, I'll just tell you that back when I was a young undergraduate, this is actually the problem that I was motivated to go off and study. Was I started off in Johnny Polka's lab looking at nervous system development in the wing of pupal flies, and there, the question is really simple. There is, you got these neurons out in the periphery of the wing, and somehow they have to send an exon deep into the animal to connect up appropriately to the ganglia of the fly. And if they don't, the fly can't sense anything with its wings. So it's all got to work together. It's this circuitry that forms by itself, no guidance, just something about how the cells interact leads to similar patterns every time. So from there, I went to the University of Oregon and I got seduced away from insects to study zebrafish. But there I was looking at the same problem, because what we found in zebrafish was they also have this repeating pattern where there's the same cells in each segment of the fish. So that was the question I was asking there is how do the cells that, in this case, we're looking at motor neurons that innervate the muscles, how do they find their way? Okay, we got some more. Oh, Fergus asked, how did the placenta evolve? Because insects don't have one. Oh, that's actually a good, that's completely off the wall, but it's a good question. Yeah, one of the key features in the early development of placental mammals is that early in development, before any of the kinds of stuff I'm talking about here occurred, uh, there's a set of cells that separate out and form just the membranes that surround the embryo. That would be another good topic for a discussion, is the embryonic membranes and what they are and how they arose. But those all emerged very, very early, and there was a core of cells that are going to form the embryo proper that don't play a role in that, although of course they interact with all those extra embryonic membranes. So how did that evolve is a good question. That there's evidence that they arose from a simpler membrane. You can find it in fish as well, the chorion, which surrounds the fish, a thin membrane that surrounds those. And we just got expansion and elaboration of that. But that there you're asking, that's a whole nother video. Okay, another question. Could you take a second to confirm I'm understanding terms about body plans and directions, specifically terms like ventral, dorsal, pectoral, anterior, and posterior? Yeah, I should have started there, I guess. Yeah, when I teach developmental biology, that's actually the first lecture, is let's get these terms straight so you know where you are in this coordinate system and where things are going. So anterior is that way, the front of the animal. Posterior is the back of the animal. It gets a little complicated in humans because we've taken our skull and we've rotated it far forward. So anterior would be my nose. Okay, and then that axis wraps around. It's also complicated because posterior would refer to the tail, not to the legs. Then there's ventral and dorsal. So in us, ventral is this side, belly side. And dorsal is backside where the spinal cord is. So that's dorsal. Let's see, pectoral. I didn't use the term pectoral. Pectoral refers to the subset. So this is, shoulder muscles, that's pectoral muscles. Yeah, I think I covered all the ones you mentioned. Yeah. So limbs, for instance, are kind of ventral structures, but limbs also have a dorsal side. This is the dorsal side of my arm, and a ventral side, which is the palm side. So yeah, there's a whole compass of axis orientations that matter. All right, another question. Could we develop without gravity? Yes, probably. Haven't tried it with people yet. They've tried it with other organisms. Fish and frogs, for instance, develop just fine in orbit, so they don't need it. There are organisms where gravity plays a role in setting things up. It's an active question about, I don't know enough about the details with the frog experiments, but frogs do depend on a gravitational displacement of yolk to the ventral side and clear cytoplasm to the dorsal side of the egg or the anterior. Oh, it gets complicated when you start referring to eggs. So in a frog egg, imagine a beach ball. The top would be anterior. That's where the nose is going to form. And then the axis is going to form along one side to the ventral side down below, which is also where the blastopore is going to form. So gravity may play one role in the formation of the egg, the initial layout of the egg, but other organisms don't care. How do they know up from down? Yeah, okay, Darren's right. Yeah, there's no such thing as up or down in absolute terms, only relative terms. That's a good answer. Yeah. So all of that is defined relatively. No, not Einstein. This is different. So, for example, I mentioned how in a frog you get this beach ball shaped thing, and there's one side that's going to form anterior. That's where the nose is going to form. And where the axis forms, that is the dorsal side, can be anywhere around there, around the circumference. But there are interactions that specify where it's going to form. In zebrafish, for instance, there is a place where the sperm enters, and that gives you another axis. So you get anterior, posterior, sperm comes in, that defines the dorsal side of the animal. Oh man, if I'm going to get into this, I should have a ball that I could scribble on to make this clear. Yeah, and then in zebrafish, for instance, all these tissues are going to migrate down and around and into the interior, and then the tail bud is going to, it's not an absolute thing. It gradually shifts as time proceeds and tissues move and all that cool stuff. Okay, somebody came in 30 minutes late, and this was a 30-minute talk, so you missed everything, but I can still answer any questions. Yeah, it's messy stuff. I literally do bring a ball into class to do this sort of thing and illustrate all these different points, because it's three-dimensional. It's things moving on a sphere. Can a sperm enter anywhere? Not necessarily. In humans, it seems to be able to enter anywhere. But in other organisms, there is a structure associated with the sperm entry point that may limit where the sperm can enter. But even in those organisms, you can get polyspermy. So it's like the egg is sitting there saying, hey, I'd like you to come in right here. This is the best spot to enter me. And sometimes, usually sperm will enter there. And sometimes they'll just say, no, I'm going in this way. Yeah, sperm can get kinky, I guess. Okay, any other questions? All right, well, I'm sitting here in my spider sweater. I actually had my scarf on earlier because my office is very cold today. The heater broke in here, and it's Minnesota, so I may have to get out of here. Yeah, who said props are used for teenage children? They were probably wrong. Yeah, I know. Like I say, when I teach developmental biology, I really rely on having those props and actually my job talk here at UMM 20 years ago, I had a couple of props. I came in with a ball to illustrate the orientation of the zebrafish embryo, and I also came in with a stocking cap, because there's a set of movements of cells called epibole in zebrafish, and you can illustrate it by putting a stocking cap, a toque for you Canadians, over the top of the ball and spreading it around. That's the kind of migrations that the tissues make. So yeah, it's when you're dealing with developmental biology in particular, you're dealing with three-dimensional movements, and it can get complicated to explain. The nervous system is often a little easier because you're dealing with a flat plane. The grasshopper nervous system that I showed you earlier, that is a two-dimensional sheet, which then acquires a third layer as cells bud off and move into the interior. But it means that you can actually make a nice little map. I showed you that diagram from Chris Doe. It's beautiful stuff where, yeah, he could line up all of his embryos and see this pattern repeating segment after segment after segment, which is kind of cool. It's like you're playing a video game. It's using textures to map things out, and if you look closely, you start to recognize the repeating texture pattern. Yeah, you see that when you start getting really intimate with insect nervous systems and seeing the pattern emerging there. Yeah, props are good at math too. Good in developmental biology. Don't use them too much in neurobiology. Oh, rubber bands for recombination. Oh, how do you do that? I don't know how you do that. Because with recombination, you got to break them, right? Huh. Yeah, in cell biology, I do try to think of ways to do demos of mitosis and meiosis and how things sort out. Yeah, I'm going to have to do that in genetics soon too, because it's kind of central to understanding recombination and mapping experiment and all that. You got to know, got to have it in your head how those tetrads break up and separate. But yeah, if anybody's got good ideas for demos you can use to show off how mitochondria behave during these processes, that would be great. Different colors. Oh. I'm going to have to contemplate this. As I mentioned, I'm going to be doing this really soon in genetics because genetics, the first thing I talk about is basics of mitosis and meiosis. Then I get into the history of Mendelian genetics. It's kind of a historical approach because people understood mitosis and meiosis before Mendel figured things out. And it was when Mendel figured things out and you brought those two together that it was, aha, now it makes sense. Those work together so well. Okay, yeah, pedagogy discussion. This would be another good thing to do with YouTube is get a bunch of teachers together and just talk about how do we teach these things. Okay, well, I'm going to wrap it up there. A little more than half an hour. That's perfect. I don't get a lot of views of these Evo Devo sciencey sorts of things. So I've been trying to keep them shorter and pithier. So short and pithy, right there. We're done. Let's move on. So I will stop right there. I'll do this again next week. You may have noticed that last week I did it at noon. Today, do it at 3, and I think I'll stick to doing it at 3 every Wednesday because I am now teaching again. So yeah, I got to work around the primary schedule, which is my teaching load. Okay, talk to you all later. It's been fun.